So these are HPLCs, high pressure liquid chromatography. We process the samples that are collected in the field, the juice, the molasses, and even the sugar with the HPLCs to separate the components and count up how many sugars are present of the sucrose, fructose, and glucose. You can also do compositional analysis with these by extracting all of the components of the bagasse into a liquid, running that liquid on the HPLC, which will separate the components out, allowing them to be counted as well. So you can determine the cellulose content, lignin contents, and hemicellulose as well. By separating all of the components into basically individual bins, you're able to count up how many of each particular type of sugar, hemicellulose, or lignin was present in the original cane star. And once you run a little bit of math and do some calculations, you come up with the total numbers that tell you exactly what each variety of cane or how many stalks had X amount of sugar, had X amount of hemicellulose. So you get a picture of all the components, what one individual stalk would contain. Sugars can be fermented in order to make ethanol, and that ethanol can be used for a fuel source or an alternative fuel. By knowing how many sugar is available for a fermentation process, we'll be able to better estimate how much ethanol can be produced off of the acreage that the sugarcane or energy cane sorghum is planted on. So you'll be able to get very good estimates of the amount of yields of ethanol you'd be able to produce off a certain amount of land. By using an alternative fuel like ethanol, we're able to offset the amount of petroleum we need to use as a fuel, reducing the amount of petroleum we have to bring into the United States for our fuel.